Hello, everyone. Thank you so much for joining us today. My name is Sophie Huckabee. I am the staff liaison for the SGNA Special Interest Groups. So if you have any administrative type questions, I would be the person to ask. Today's webinar will be presented by Lisa Brown. She is the chair of the Associates and Technicians Special Interest Group. And I will turn the webinar over to her now. Thank you so much.
I'd like to thank you for attending. I'd just like to tell you a little bit about myself. My name is Lisa Brown, and I've been an endoscopy tech for about 12 years now at the University of Virginia Medical Center, and I belong to the Old Dominion region in Virginia of the SGNA. So let's get started with our webinar. Today we're going to talk about basic anatomy and physiology of the GI system for the GI tech. This is just a picture of the GI system. This is what we'll be talking about tonight. Our objectives are the basic knowledge of the GI system and the anatomy, the basic knowledge of the GI system physiology, and to be able to identify the GI system, the landmarks. This is just the structures of the esophagus. The walls of the esophagus is made up of 3 layers, the mucosa, the submucosa, and the muscularis. The esophagus is a hollow muscular tube approximately 23 to 25 centimeters in length and 2 to 3 centimeters in diameter in adults. The esophagus serves as a channel for food to go through the mouth to the stomach. It's considered the 3rd organ of the digestive system after the mouth and the pharynx. It's located posterior to the trachea and the larynx. It passes through the diaphragm into the abdomen at the diaphragmatic hiatus. Almost immediately after the esophagus, it passes through the hiatus and enters to the stomach. At the end of the esophagus is the upper esophageal sphincter. At the lower part of the esophagus is directly above the angle of the His, and at the junction between the tubular esophagus and the stomach is the lower esophageal sphincter. The motility of the esophagus, at rest, the upper and the lower both are closed. When a bolus of food is pushed through the pharynx, the upper sphincter opens, allowing the bolus to pass through the esophagus. At the end of this point, the gravity and the peristalsis comes to advance the bolus down through the esophagus. The peristalsis is defined as distally progressive band of circular motions at the rate of 3 to 5 centimeters per second, effectively transporting the material toward the stomach. The lower sphincter controls the passage of the ingested food into the stomach. The bolus reaches the lower sphincter. It opens and allows the bolus to enter the stomach and then closes to prevent the reflux of the food and the acid back into the esophagus. This is the regions of the stomach. The stomach consists of 5 different layers: the serosa, the subserosa, the musculus propria, the submucosa, and the mucosa. The stomach is J-shaped. It's a distal organ. It is located just between the diaphragm, above the esophagus, and below the duodenum. It is approximately 25 to 30 centimeters long and 10 to 15 centimeters wide at its widest point. The portion of the stomach that immediately adjoins the esophagus is the cardia. The gastric fundus is a dome-shaped part of the stomach that extends to the insula angularis, a notch-like indentation located on the upper lateral border of the stomach. Below the insula angularis and extending to the narrow tubular pylorus is the antrum. The upper lateral border of the stomach is the lesser curvature, and the lower lateral border is called the greater curvature. The stomach has several important physiological purposes. It maintains the relatively low microbes in the upper digestive tract. It digests and prepares nutrients for absorption. It serves as a reservoir for swallowing food, drink, and digestive secretions, mixing and delivering the chyme to the small intestines for further digestion and absorption, and it originates the signals for hunger. The entry of the food into the stomach is controlled by the LES, which comprises of a group of thickening circular muscles at the distal end of the esophagus. The LES regulates the opening and closing of the esophagus to the lumen, the esophageal lumen. At the distal end of the stomach is the pylorus, which is a thick muscular wall that forms the pyloric sphincter. The pyloric sphincter controls the movement of the stomach, the contents into the duodenum. The layer contracts to produce the peristaltic motion of the stomach while it churns and compresses the food during digestion. The gastric mucosa lines the interior of the stomach. When it is not filled, the stomach interior has wrinkles of muscles called the rego. The rego allows the stomach to distend, hold large quantities of food without any substantial increase in pressure, thus helping to control the rate of the food that enters the duodenum. The primary function of the stomach is to initiate digestion by using both controlled secretions and mechanical movements. In addition to, the stomach serves as a reservoir for ingested food and liquids and regulates the entry of food into the duodenum. The stomach contains three types of glands: the cardiac, occipital, and the pyloric. These glands secrete different substances that break down the chyme. When the chyme is at rest, normal secretions occur at the rate of about 0.5 milliliters per minute. With food in the stomach, the secretions increase to about 3 milliliters per minute. The stomach secretes approximately 1,500 milliliters to 3,000 milliliters of gastric juices daily. The motility of the stomach. The movements of the stomach serve in two of the following functions. Mixing and grinding, which takes place in the distal areas and is controls the emptying of the gastric contents into the duodenum. The peristaltic movement of the stomach combines each bolus of food with a mixture of digestive juices to form relatively homogeneous semi-liquid masses called chyme. The peristaltic movement of chyme slowly moves toward the pylorus at the rate of about 3 waves per minute. Before the chyme is ready to enter the duodenum, it must be the proper consistency, and the duodenum must be receptive to the bolus of food. When these conditions are present, the antral peristaltic contractions force the chymes through the pyloric sphincter into the duodenum. Only a portion of the chyme is advanced toward the pylorus, actually moves through the pyloric sphincter into the duodenum. The rest is propelled backwards, colliding with and further breaking down large particles in the antrum and the body. Particles passing into the duodenum have a diameter of 1 millimeter or less. The gastric emptying rate controlled by neutral impulses by composition of the chyme and the hormones secreted by small intestines. This is the regions of the small intestines. The small intestines is a tubular structure that begins distal to the pyloric sphincter of the stomach and extends to the cecum of the large intestines. It has digestive, absorptive, secretory, barrier, and immunological functions. The small bowel is approximately 5 to 6 meters in an adult. The first 30 centimeters of the small intestines is a C-shaped muscular duodenum, which begins at the pyloric sphincter and at the end and ends at the ligament of Trice. The common bile duct empties into the duodenum at the ampulla of Vater. After the duodenum, the proximal 2/5 of the small intestines is known as the jejunum. The distal 3/5 of the small bowel is known as the ileum. It extends from the duodenum to the ileocecal valve. The valve controls the flow of chyme into the large intestines and prevents the reflux into the ileum. The mucosa and the submucosa are arranged in circular folds, which provide a greater surface area for secretions and absorption. During the 3 to 6 hours, the chyme remains in the small intestines. The mucosa forms an estimated 3 to 5 million small finger-like villi which project into the intestinal lumen. The primary function of the small intestines is to absorb nutrients from the chyme. The small intestines receive up to 8 liters of fluid per day and passes 500 to 1,000 milliliters into the large intestine. The watery fluid supplies a carrier substance for absorption of the nutrients when the villi come in contact with the chyme. There are about 10 different types of endocrine cells located in the crypts that produce a number of peptides and hormones. The motility of the small intestines. With the small intestines, there are three movements that contribute to mixing the chyme. Concentric segmented contractions, which normally take place in the jejunum and give the small intestines the look of the chain of sausage links. These movements help the mixture of secretions in the small intestines with the chyme particles. Short, propulsed contractions or the peristaltic waves slowly push chyme into the direction of the colon. These contractions are found predominantly in the first portion of the duodenum and the jejunum. The continuous shortening and lengthening of the villi constantly stir the intestinal contents. After the chyme nears the large intestines, contractions increase in the ileum. The ileocecal sphincter relaxes, allowing the chyme to move into the ileum, from the ileum into the cecum. This is the sections of the large intestines. The large intestines is about 120 centimeters to 150 centimeters long, about 4 centimeters to 6 centimeters in diameter. It extends from the ileocecal valve to the anus and is divided into 5 sections: the ascending colon, the transverse colon, the descending colon, the sigmoid colon, and the rectum. The large intestines consist of 4 layers: the serosa, the submucosa, and the mucosa. Immediately above the cecum, the ascending colon, which is also known as the right colon, passes up to the right side of the abdomen to the lower border of the liver, where it bends to the left at the hepatic flexure. The transverse colon is the portion of the bowel that crosses the abdomen from right to left, from the hepatic flexure to the splenic flexure. The descending colon, which is also known as the left colon, runs down the left side of the abdomen from the spleen to the iliac crest, where it's a S curve at the midline to form the sigmoid colon. The intestines terminates at the rectum and the anal canal. The rectum length is about 9 centimeters in infants, increasing to 15 centimeters in adults. The movement of feces through the anal canal is controlled in an inch-wide sphincter composed of involuntary smooth muscles and extended sphincter made up of voluntary stride rate muscle. The levator muscles surrounding the rectum also help keep defecation under voluntary control. The ascending and the descending colon are retroperitoneal, while the transverse and the sigmoid colon lie interperitoneal. At the distal end of the small intestines, a flap valve known as the ileocecal valve lays. It acts as a sphincter that controls the passage of intestinal contents from the ileum to the colon and to prevent reflux of bacteria back into the colon into the small intestines, thus preserving a relative sterility of the small bowel. The appendix itself is a tubular structure that ranges from 2 centimeters to 20 centimeters in length. The motility of the large intestines. The colon has 3 basic movements: periodic, uncoordinated tonic contractions. Both fondatoal and circular motions bunch up into the folds of the mucosa from the hysteria. Phasic, random, non-pulsative contractions, at least 30 seconds to 2 minutes, and include peristalsis and retrograde peristalsis. This mixes the stool material to help absorb its liquid content without advancing the material toward the anus, and spontaneous mass movements, which occur 3 to 4 times a day when the colon becomes filled and distended. The rapid movement of this material is more evident in the colon than in the small intestines in mixing the movements. When masses move out of the sigmoid colon, it moves the feces into the rectum. The urge to defecate is stimulated, causing peristaltic waves in the rectum and relaxation of the internal and the external anal sphincter. If relaxation of the internal and external sphincter is not enough to provide defecation, a Valsalva maneuver assists in the process. This maneuver is an involuntary movement of the bowel wall and the relaxation of the external sphincter, assisted by the contraction of the diaphragm and the thoracic and the abdomen muscles. The strength and the downward push lasts about approximately 8 seconds, increasing the inter-abdominal pressure and the frequency of emptying the colon from as high as the splenic flexure. Under normal conditions, gas moves down the colon at the rate of 1 to 4 inches per second. Liquid moves by gravity, peristalsis, and by movement. Overall, the motility of the large intestines is slow compared to the small intestines, and it can take up as long as 7 days for material to clear the colon. Secretions in the large intestines. Colonic secretions are scanty and consistently primary of water, mucus, potassium, and bicarbonate. Chemicals and mechanical irritation increases the secretions of the fluid. Absorption and emulation of the large intestines. Of the 1 to 2,000 milliliters of liquid chyme that enters the colon daily, only 100 to 200 milliliters of fluid is evacuated in stool. The colon is much more effective than the small intestines in removing water, operating at 80 to 90%. The bacteria in the colon. The colon is sterile at birth and rapidly becomes colonized when you swallow by swallowing bacteria. The bacterial composition is established primarily from the material contamination within 1 week of birth and is maintained throughout the lifetime. The adult colon contains approximately 109 to 1,011 organisms per gram of stool, which consists primarily of gram-negative anaerobic cocci and aerobic bacterium. Normal feces contain intestinal bacteria that help break down purified body waste, traces, fluid, mucus, indigestible cellulosis, undigested connective tissue or toxins from meat proteins, undigested fats, and bile pigment. Approximately 500 species of 1 trillion organisms live in a healthy colon. Feces are normally brown in color because of the metabolism of bile pigments to stercobilin. Odor is a result of indole and stactol, mercaptan, hydrogen sulfate, and breakdowns of products of cysteine. And all our references come straight out of the core curriculum for the SGNA.